All right, so the last lecture wanted to include this week is talking about membrane transport. Specifically talking about cellular membranes, and then some processes by which things are moved across the membrane, because it's very important for cell maintenance, cell survival, that things get moved across the cellular membrane. And as we said, cellular membranes are lipid bilayers, okay? They're selectively permeable. Some things can move across cellular membranes easily, some things can't. Right? Water can cross, but ions and most polar molecules cannot. So water is kind of a unique thing because water's polar, but most polar molecules cannot. So it kind of breaks it down there. So here are some things that can easily move across this selectively permeable membrane, right? This lipid bilayer, the hydrophilic heads and the hydrophobic tails, all right? But most things cannot. All right, now we've talked about the cell membrane being this phospholipid bilayer, but it's not just phospholipids, okay? You have proteins. You have other types of molecules. You see here that you have glycoproteins, glycolipids, cholesterol is a big component of cellular membranes. And so that's why cellular membranes are considered a fluid mosaic. Fluid because these lipid bilayers can move against one another. Okay, they can shift. And mosaic is that it's a collection of different things. Okay, so a fluid mosaic. And if you look at the proteins associated with cellular membranes, membrane proteins, you have those that are integral proteins, all right, and integral proteins are permanently attached to a membrane bilayer, and some even span the entire membrane, okay? So these right there, here's an integral protein, all right, that spans the entire length of the cellular membrane. But you also have peripheral proteins, such as these blue circles there. They only temporarily attach to one of the lipid bilayer surfaces. So it may be interior of the cell, it may be exterior of the cell, but they are only interior, temporarily attached, whereas these integral proteins are permanently attached. So like we said, water and other smaller molecules can diffuse, and we'll talk about diffuse in a minute, but can move directly across membrane protein or can move directly across the plasma membrane. So the things that can't may need the help of transport proteins. All right. So here, transport proteins can exist as a channel. All right, so you see these sort of green pentagon molecules moving down this channel. All right, or they may exist as what's basically carrier molecules. They sort of open up and carry this molecule across the membrane and let it go to the other side. But you have other types of integral proteins. So here you have transporter proteins. They exist on all cellular membranes and assist the movement of substances across the membrane. You have some membrane proteins that are recognition proteins, all right, that help function as identity tags for the cell. Then you have some membrane proteins that are receptor proteins that bind to specific extracellular substances. So we've talked about these different molecules associated with the plasma membrane, but now let's talk about the specific movement of substances. All right, so we're going to first talk about diffusion. So diffusion refers to the spontaneous movement of ions or molecules and has important implications for cellular functions. All right. So a basic example of diffusion is you have this beaker full of water and you drop a couple of drops of food coloring. All right. And that food coloring or those food coloring molecules spontaneously and randomly diffuse into that water. Okay. And so eventually it's kind of more spread out. All right, and so the rate of diffusion, or how quickly diffusion happens, is influenced by a number of factors. Size, okay, molecular size. Molecules that are smaller in size diffuse faster than larger molecules. Okay. Temperature. Higher temperatures increase the rate of diffusion because molecules are moving faster. All right, so think about a cold day versus a warm day. On which day do you move faster? Okay, usually the warmer day. Same thing with molecules. The warmer the temperature of the environment they're in, the faster they're going to move. Charge is another factor that affects the rate of diffusion. Oppositely charged substances will diffuse quicker to each other than non-charged substances. So if you have two things, one a positive charge, one is negatively charged, and they're diffusing to one another, they're going to diffuse quicker to each other than molecules that are uncharged. Pressure is another factor. The higher the pressure that is exerted on the molecules, the faster rate of diffusion. And then finally, the concentration gradient. All right, so molecules will move from an area of higher concentration to an area of lower concentration. All right, and so if the concentration gradient is larger, they're going to move faster. All right, so if you have a lot of molecules on one particular side of a membrane and very few molecules, all right, the molecules that are a lot more are going to diffuse across the membrane to the area where there's fewer molecules there. Okay, so they're moving down their concentration gradient. All right, if you have two situations where you have the molecules are a little more equal on each side, right, that concentration gradient is lower. So they're not going to move as fast, okay, from high to low than if it's a whole bunch of molecules on one side and just a few molecules on the other. And then they're going to diffuse a lot quicker. All right, so now let's talk about osmosis. Now, osmosis is specifically talking about diffusion of water across a selectively permeable membrane. Now, when we're talking, we've been talking about molecules moving across a membrane. Okay, well, usually those molecules are dissolved in water. So you not only have to think about the movement of the molecules across the membrane, you have to think about the movement of water across the membrane. Okay, so osmosis occurs in response to a difference in solute concentration. And remember, the solute is what's dissolved in water. Okay, and so osmosis will occur in response to changes in solute concentrations, all right, or tonicity, okay, between the fluids on either side of the membrane. All right, so there's three terms that you really want to pay attention to. So in an isotonic environment, okay, the solute concentrations of the fluids on both sides of the membrane are pretty equal. There's no net movement of water, no osmosis. Okay, there's also no net movement of the molecules of the solutes. All right, in a hypotonic situation, let's say you have a cell that's placed in a hypotonic environment, then the environment has a low solute relative to another fluid. If it has a low solute, it means it has more water. Okay, All right. And so in that case, the water is going to go from the environment into the cell. All right. Now, let's say you have the cell, that the cell is hypotonic, meaning that there is fewer solutes inside the cell than outside the cell. And if that's the case, you have more water inside the cell than outside the cell, so water is going to move outside the cell. Okay, and then finally, hypertonic. All right, let's say you have a cell that's hypertonic to the environment. That means it has more solutes inside the cell than outside the cell. All right, so where are the solutes going to go? They're going to leave the cell. Okay, where's the water going to go? It's going to go into the cell. Okay, all right. Now, if you put that cell in a hypertonic environment, the exact opposite is going to happen. Okay, in a hypertonic environment, what happens? There's more solutes outside the cell than inside the cell. Okay, so where the solutes going to go? They're going to go into the cell. Where's the water going to go? It's going to go outside the cell, and the cell is going to shrink. All right, so here you have two environments, okay, separated by selectively permeable membrane. The red dots represent the solutes inside, dissolved in the blue water. All right, now on which side is the solute concentration higher? This side or this side? Well, it's this side. You have more solutes, so the solute concentration is higher on this side. All right. Now, if the molecules, if the solutes, these red dots, can't move across this membrane, but the water can, where's the water going to go? It's going to go from an area of higher concentration. Now we're talking about the water, not the solutes. It's going to go from an area of higher concentration to an area of lower concentration. And that's what you see here. That water is going to go across that selectively permeable membrane to this side of the environment. Okay, all right, to try to even out the solute concentrations on both sides. All right, now when we're using these terms, I try to be specific is whether I'm talking about a cell or the environment, because these are relative terms. All right, and so in this slide, we would say this cell, okay, is placed in a hypotonic environment or hypotonic solution. All right, again, that means that there's more water in solution or in this environment and then there is in the cell. All right, so where is the water going to go to? It's going to go into the cell. All right, as opposed to hypertonic solution, more solutes outside the cell than inside the cell. That means you have more higher concentration of water inside the cell than outside the cell. So where's that water going to go? It's going to go outside the cell. All right. So in this situation, what's going to happen to the cell? If water leaves, it's going to shrink. In this situation, water comes into the cell. So what's going to happen in the cell? It's going to swell, and it may even burst or lyse. Okay. So in an isotonic solution, they're pretty much equal. So you may still have some movement of solutes or water, but it's going to be pretty much equal. So there's no net movement of water. All right, so like we said, just mentioned there, you need to be able to understand what happens to a cell in these different environments. All right, so if this is a cell right here, it has 2% sucrose, which is a solute. All right, if you place it in a 2% sucrose solution, nothing's going to happen. It's going to stay the same. If you put it in a 10% sucrose solution, what are you saying? What type of environment is that? Well, the solute concentration is higher outside the cell than inside the cell, so you're putting it in a hypertonic solution, so water's going to leave the cell and it's going to shrink. Okay, now you're putting in a water solution, so basically it's 0% sucrose, so the water concentration is higher outside the cell than inside the cell, so water's going to move inside the cell and it's going to swell. All right. So these are pictures of red blood cells in these different situations. Okay. All right. So this is what your red blood cells should normally look like. So this means they're in a hypertonic solution. Here, I'm sorry, isotonic solution. And C, you see they're shriveled up, they're losing shape, getting smaller, misshapen. So that means they're losing water. So what type of environment have they been placed in? A hypertonic solution. Here, all right, they're becoming big red balls. That means water is moving into the cell. In fact, you can see some that have lyse, meaning burst open. So what type of solution were these placed in? A hypotonic solution. All right, now animal cells, all right, they prefer isotonic solution. All right, they don't like hypotonic because they burst. They don't like hypertonic because they shrivel or crenate. So they prefer isotonic solution. Plant cells are different. They prefer this hypotonic solution. Why? Because that the water, incoming water, is going to go into that central vacuole that we talked about in the cell lecture, and that central vacuole is going to push up against the cell wall and maintain what we call turgor pressure. Okay, and so when you look at very healthy looking plants, their stalk is straight, and the leaves are out wide. That means that the plant is maintaining its turgor pressure. If it starts to wilt, it means it's losing water. All right, then those cells are starting to shrivel a little bit. So here is under hypertonic solution versus hypotonic solution, good turgor pressure. So plant cells prefer a hypotonic solution. Now let's talk about the energy needs of different types of ion or molecule transport. Okay, when energy is not needed, it's called passive transport. So passive transport does not require the input of energy because the movement of molecules is down the concentration gradient. It's going from an area of higher concentration to an area of lower concentration. It's going down that hill. All right, and if you are running down a hill, you don't need a lot of energy. You let gravity take the lead. Okay, so molecules going down the concentration gradient don't need energy to move there, so it's passive transport. Now, if a membrane protein or something is needed to facilitate diffusion, okay, it's called facilitated diffusion, but it's still passive transport because energy input is not needed. However, there are times where the cell needs to move molecules or ions against their concentration gradient from an area of low concentration to an area of high concentration. All right, and so those molecules or ions are moving up that hill from low to high. And if you go up a hill, you need energy, right? It's much easier to go down the hill, but when you go up it, you need energy. So to go against a concentration gradient is considered active transport because you need the input of energy. And the most common way a cell generates that energy for active transport is through the use of ATP. Now remember, we mentioned ATP in the organic macromolecules lecture. Okay, what type of macromolecule is it? It's a nucleic acid, adenosine triphosphate. Well, the cell will take one of those three phosphate groups and cleave it or take it away from ATP to make it ADP, adenosine diphosphate. And that simple thing of removing one of those phosphate groups releases energy. All right, and so the cell can use that release of energy for active transport to move things against a concentration gradient. So a classic example of this is a sodium potassium pump. All right, and so not really need to talk about the purpose, all right, or specific examples of what this is used for. You'll talk about this in other classes, all right, but this is a classic example of active transport because this membrane protein moves sodium against its concentration gradient outside the cell with the help of ATP, and it moves potassium against the concentration gradient inside the cell. Okay? Again, active transport and needs the input of energy in the form of ATP to move things against a concentration gradient. Now, some things are moved across are moved across plasma membranes, but they're larger, and they are removed or taken in through the use of vesicles, and which are membrane-bound organelles. All right, and so you have vesicle movement. You have exocytosis, which means cell expels the vesicle's contents to the outside environment, to the extracellular fluid. All right. Then you have endocytosis, which means the cell will take in a small amount of extracellular fluid and whatever is in that fluid, right, by the ballooning inward of the plasma membrane. Now, there are two types of endocytosis. You have bulk phase endocytosis and receptor-mediated endocytosis. So with receptor-mediated endocytosis, basically a specific molecule binds to a receptor, okay, hence the name, and that will signal the cell membrane to balloon in, and eventually it will pinch off here to form this coated vesicle, all right, with whatever materials that was in this extracellular fluid that was taken in. All right. You have endocytosis if the cell eats something, quote unquote, eat something, takes something in. Now we see this with our immune system, phagocytosis, where a phagocyte, immune cell, takes in some foreign cell and digests it. You also have penocytosis, which is cell drinking. Now, exocytosis, all right, you have a vesicle that has something that the cell needs to get rid of. All right, well, this vesicle will bind to the cellular membrane, okay, and then open up to expel whatever inside to the external environment. But as you can see, what makes up the membrane of the secretory vesicle? These phospholipids, okay? So you have these phospholipids that were once part of the secretory vesicle, now will become part of the cellular membrane. All right, so exocytosis adds to the cellular membrane. Endocytosis takes away from the cellular membrane. So membrane proteins and lipids are made in the endoplasmic reticulum and then moved to the Golgi bodies for final modification. And these vesicles, which are finalized, contain proteins and lipids, and they move to infuse with the plasma membrane. All right, and so exocytosis and endocytosis, like we just said, continually replace and withdraw parts of the cellular membrane. Exocytosis adds to the cellular membrane. Endocytosis takes away. It's just, so here's an example of phagocytosis, All right, some bacteria that's foreign to the body, and this immune cell takes it in. All right, so that's the end of this lecture. So here's the need to know slide. So how do cell membranes regulate movement across them? Okay, what is diffusion and osmosis, and what factors regulate these processes? What are the three types of tonicity that we talked about, and which tonic environment or tonicity do animal and plant cells prefer? All right, what's the difference between passive and active transports, and what are types of vesicular transport and their characteristics? All right, again, let me know if you have any questions about material that we covered in this lecture.